Okay, now for this part, I want to talk about how that initial filtrate that's made up in the renal corpuscle gets modified into urine. This is mostly the job of the tubule system in, well, working with the peritubular capillaries or the vasa recta in different parts. We're also going to talk about how countercurrent exchange works to be able to concentrate up the urine. We're going to focus most of our discussion talking about salt and water, though we'll talk about nutrients and urea a little bit around the edges. But this is kind of a complicated story, but we'll kind of work our way through it. So we could summarize that remember that the filtrate that comes out of the renal corpuscle is basically the same constitution as the plasma is. Okay, so whatever nutrients or wastes or salts or almost anything else except for plasma proteins that are in it, it's basically got the same kind of concentration when it gets there. However, in blue, here you can see there are a lot of substances that will be reabsorbed, from the filtrate to go back into the plasma, and others that are going to get secreted actively from the plasma to go back into the filtrate and eventually turn into urine. Now, remember, this is cells of the tubule system that are actually doing these processes, though, so there are a couple of different mechanisms that we can identify. This is a little bit of an older picture than the ones in the current version of the textbook, but let me walk you through it. So on the left part of this diagram, here's the filtrate and the lumen. Here are the cells of the tubule here, an exaggerated interstitial space, and then the peritubular capillary that's going around the tubule. So what we see is two kinds of ways of movement. The first one is called transcellular movement. Notice how transcellular movement goes like right through the cell from the apical surface and gets pushed out its basilar surface. And there are both passive as well as active, in red, routes that different substances and water can take to get their way out. Additionally, and this tends to involve transporter proteins or pumps or the like. On the other hand, there is a certain amount of passive leakage that can happen through the peritubular capillary into the peritubular capillaries as well, through what's called the paracellular route. So going between the cells rather than like pumped through them in order to get out to the peritubular capillary. There are tight junctions, which you remember are permeability barriers between these cells, but particularly the proximal convoluted tubule is actually quite leaky, so it's going to allow a lot of substances out that way. So, for example, if we looked at how this process happens in the proximal convoluted tubule, there are a series of different maneuvers that happen. And I want to talk a little bit about how these relate to other, some of the hormones that we've talked about before. So this kind of starts arbitrarily here in looking at this sodium-potassium pump. This is like the same old 3 sodiums out for every 2 potassiums in kind of pump that we've been looking at, but it's associated with this kind of passive, facilitated diffusion here, where there are these leakage channels that allow the potassium to kind of cycle through. Some of the potassium is going to get returned to the plasma, but the rest of it's just going to get shuffled right over to the pump. So what happens is there's, through active transport, so secondary active transport here, sodium gets moved through along by way of a transporter protein, and then it's actively pumped out here. So basically, the sodium concentration of this cell goes down, which is what draws in the sodium from the filtrate, and then the filtrate actively gets pumped uphill to go on its way out. There are other substances that are in high concentration here, like these foodstuffs that are indicated, or vitamins, that are also going to kind of move their way through and can also get basically going down their concentration gradient to get outside. There are structures here. Structures. There are proteins on the surfaces of these cells that are called aquaporins. Now, an aquaporin is a channel that allows water to roll down its concentration gradient. You remember antidiuretic hormone from back in the old days? Remember, ADH is what causes you to make a concentrated urine? Here's how it happens. Antidiuretic hormone causes genes to get turned on that make these aquaporins and put them on the surfaces of these cells. When there are more aquaporins present, that lets water go from here, in the filtrate back into the peritubular capillary. When there are fewer aquaporins on the cell surface, that makes it harder for water to go through the cell and the way out, and basically you're stuck with just kind of the leakage that happens on its own. So it's ADH that makes these aquaporins that causes the dumping out of water. And again, water, if you're moving lots of salt, the water will follow the salt on its way out. In addition to this, the hormone aldosterone that we talked about before, that's actually what makes the sodium-potassium channels. It causes the cell to produce more sodium-potassium channels, put them on their apical surface. So there reaches, at a certain point, what's called the transport maximum. And actually, this transport maximum goes for any kind of substance. Remember I said before that, it's rare that you're going to find glucose in a healthy person's urine for the reason that that's like wasting this energy. If someone is diabetic, there's going to be a lot more glucose in the bloodstream, in the plasma, therefore lots more in the filtrate. However, there comes a point when there can't be any additional, basically you run out of either room or capacity to be able to make transporter proteins for them. But basically, if you lack channels in order to be able to move glucose through these cells, glucose will end up getting left over in the filtrate and then pushed out in the urine. So basically, those are two things I want to have in your head. The idea that sodium can be actively moved by emplacing pumps. Water is allowed to follow sodium or to roll down its concentration gradient by the emplacement of aquaporins due to ADH. But lastly, every substance, no matter what it is, has a transport maximum, past which point, once you've got a lot of it in the filtrate, there comes a point where you just can't reabsorb it all. This process happens in different places in the tubule. I'm not going to get into a lot of detail here in this video about exactly where it happens, but this is like the big picture of how these substances get moved. Now, I want to move on to talk about how the urine gets concentrated. Now here we're largely talking of the story of how you either, depending on your point of view, either pull water out of the urine in order to make it more concentrated, but what also happens is salt will also get pumped in, will be allowed in or out too. Now, so generally the thing to know is that the salt concentration of the plasma is about 300 milliosmoles per milliliter. So you see these little 300s up here in the cortex. However, as you go deeper in the medulla, notice that the solute concentration of salt in this case goes way higher, up to 1200 milliosmoles. Now, the reason for this is based on the thing that we did in lab, where it's a countercurrent flow system. By way of reminder, remember, in most cases, if we are talking about flow, say moving from the left side of this pipe to the right side, imagine this is oxygen, and so the artery would be here. If the artery starts off with a high partial pressure of oxygen, by the time it gets to the end of the capillary bed, it's less oxygenated. Again, not fully deoxygenated, but it's reached equilibrium, basically, with the cells around it. This is sort of an arbitrary example that I totally boosted from Wikipedia, but the thing to dig about it is that if we were doing partial pressure, this would be like close to 100 millimeters of mercury by the time when it leaves the capillaries in the lung. But then by the time it's done doing its exchange, at the end of the capillary bed, it's going to be close to like the 40 millimeters of mercury that the plasma was initially, that the cells were initially. So it's going to help. This is going to become more evened out as it goes from one side to the other. Shown in graphical form, and this is for Gill, but still it works the same for what we're talking about, where if you start off with the substances moving in the same direction, if one's got a high concentration of something, gas or solutes or something else, and one's got a low concentration, as you go from one side of a capillary bed to the other, they're going to reach some kind of intermediate value. Now, contrast that to the example that we gave before with countercurrent flow. When you have, in this case, the upper pipe is moving right to left and the lower one's moving left to right, it allows you to build up these gradients where just by using diffusion rules, if you move the pipe, if the flow is going in opposite directions, near one end of the pipe, it's going to be more concentrated, and at the other end of the pipe, it's going to be less concentrated. Unfortunately, the heat map doesn't totally work between this figure and my other figure, but bear that in mind because those are again like the ground rules for how this happens. Now, this gets a little bit tricky in the way that it's explained in the book, so just sort of tag along with me as I explain this. There are a series of pictures in your text that kind of outline this that I'm going to walk you through now. First, the big picture here is that the blood that comes out of the efferent arteriole and goes into this Vasa recta, again, this straight vessel, it's going to tend to be basically like more highly oxygenated up here, less highly oxygenated there. But these cells are actually running parallel to one another. But there's actually a problem with this figure that I'm going to talk about a little bit later. Here's something that I want you to remember. Remember that as the filtrate goes down, it goes down the loop of Henle. As it goes down through the nephron's loop, water leaves passively. Okay, so the descending limb of the nephron's loop is permeable to water. So as water goes, as the filtrate goes down and water leaves, that's going to cause it to get more concentrated. Then, as it comes up here in the ascending limb, it's now impermeable to water, but it also pumps out salt. So here it's becoming, but it pumps out salt. So on its way down, it's becoming more concentrated because it's losing water. On the way back up, it's becoming less concentrated again because it's actively pumping out salt. So in the end result is, so the salt's pumped out at the end, which means the interstitial fluid around those cells has a high osmolality, meaning it's got a lot of concentrated solutes outside. As a result, on the descending limb, water leaves to try to neutralize this, right? Water is going to be in relatively low concentration, so it's going to try to, through osmosis, try to be able to even it out. Then it becomes, which then is going to increase this osmolality of the filtrate as it goes down. Filtrate, as it continues along, it's more concentrated as it goes up to this limb, then salt's pumped out. Salt's pumped out, so that's going to increase the salt concentration of the interstitial fluid. On this side, because there's high salt outside, water leaves. So again, and as a result, the filtrate becomes more concentrated as it goes down the bottom. So what this cycle does is it has the effect by passively allowing water to leave and actively pumping out sodium chloride. It's going to have the effect of having a more concentrated filtrate down here at the bottom of the hairpin than it is on the other side. So if you look at, and this is actually a helpful figure, if you look what happens is, so it starts off at 300 milliosmoles in the plasma, and so is the filtrate. Goes through the proximal convoluted tubule, and then water starts leaving passively because there's like a lot of sodium outside. See, the sodium is getting moved out. So a lot of sodium outside, so water leaves. Water leaves. The filtrate starts going from 300 to 400, 600 to 1200 down here at its deepest when we're talking about a juxtamedullary nephron. And then as it goes on its way out, water is able to leave a little bit here too. But now sodium starts actively getting pumped out. Sodium leaves passively here because it's in high concentration. Then it starts getting pumped out at the same time, making the interstitial fluid more salty, which also draws out more water. But by the time the filtrate gets all the way out to the distal convoluted tubule, notice how this is now much less concentrated than the initial filtrate was. Now, this might seem like an exercise in futility, but it'll make sense why this is done, like for a functional reason, in a little bit. But just sit tight for that example. For that explanation. This view here of what's happening with the Vasa recta, again, those peritubular capillaries that are going around the nephron's loop, that is allowing water and salt to like enter passively throughout this whole route. But it mirrors what's happening with the concentrations, where it's starting off more concentrated, where it begins, and then getting less concentrated as it ends up on the venular side. Now, here's the part that I think is not right and has really not been fixed. If you have an older edition of the book, this is one of the figures that's used to explain it. So this looks like it's just the two preceding slides kind of all put on the same thing. But here's what's the problem. The problem is we see the filtrate going down the hairpin and then up this way. And we see the blood going down the hairpin and then up this way. My issue with this is we're supposed to be doing countercurrent flow, right? So you think this should allow, now we're seeing concurrent flow because they're both having like the same fluid pattern. So the way that I kind of did a crappy drawing of this, and so the way to think about it is not that the blood's moving in the same direction. The blood's not moving in the same direction as the filtrate. So for example, if I drag this guy over here, you can see the way that these are now overlaid. The filtrate is the filtrate's going down on the descending limb and up on the ascending limb, but the water is moving in the opposite direction. The plasma is moving in the opposite direction through the pipe. But then how is this going to work? So now I made actually a second one here. So if I put them like this, they start to kind of if they kind of overlap one another, I guess like this is okay. When they start to overlap one another, now you can sort of see how this goes. Now this looks kind of a jumble, but see those like cut surfaces I have here on my on each of these pipes. Remember what the medulla of the kidney looks like. This is a micrograph of the medulla of the kidney. The slightly larger openings, those are collecting ducts. Everything else you see are ascending and descending loops, limbs of the loops of the nephron's loops, and the Vasa recta. You kind of can't tell which is which on this particular slide, but the take-home message is, how can it be that you have, continue to have fluids moving in opposite directions to do countercurrent flow? It's because all of these pipes are like right next to each other and kind of jammed together. Well, anyway, the big story is, so again, if you look at this picture, this gives you the impression that the filtrate gets wicked concentrated and then really dilute again. However, it's almost like you never be able to concentrate urine. But as it turns out, where the real action is for concentrating up that final urine, that happens within the collecting duct. So the collecting ducts use the same gradient again. So now, this is actually, I think, a really helpful picture. So this is on the left, this is if we were so overhydrated that we had no ADH. If you had just sucked down a big 64 ounces, big gulp or something, what happens is the fluid that's isoosmotic with the plasma starts off at 300 milliosmoles and then gets more concentrated up to 1200 and then gets more dilute again. Now the thing is, when you have that much extra blood pressure because of the additional fluid you've taken on, that's going to cause antidiuretic hormone to get shut off, right? So when you shut off antidiuretic hormone, that is going to prevent the emplacement of the aquaporins in the cell membrane. So you remember, aquaporins up here are these ones that allow water to move down its concentration gradient. So if we're overhydrated, we have no ADH, no aquaporins in the collecting duct, which means you don't lose any water. You'll cycle a little bit of this urea just to kind of keep the countercurrent system going, but you end up with a large volume of dilute urine, which is the problem that you're trying to solve. Too much water on board, that's how you excrete it. On the other hand, if you were either dehydrated or just had like a super salty meal that you need to be able to dump out the salt from, it starts off the same way, where you concentrate up the filtrate through the bottom part of the loop, then it gets diluted again, However, because ADH is used here, because you want to concentrate the urine, ADH will emplace these aquaporins, which will allow water to leave passively. So by the time it gets to the bottom of the collecting duct, it'll be the same concentration as the bottom part of the loop. All right? So this is the difference now. If when there are aquaporins present, because you're trying to concentrate up urine, then the urine can get concentrated as on the right. On the other hand, when we're overhydrated and we don't have any ADH, then those aquaporins are going to get pulled off those cells, like not retracted, but like there are mechanisms in the cell that'll cause them to get removed from the cell membrane. Not going to allow water to move passively again. So it's really the collecting duct that makes the choices about where water is going to be, whether water is going to be removed from the filtrate or kept in the filtrate. That's most of what I wanted to get through for today. So we'll unpack this in lecture. See ya.